Hey everybody, and welcome back to Blackboard Biology. So in our previous videos, we have looked at DNA, not super in-depth, but in-depth enough for a high school biology class. And so now it's time to turn our attention to, you guessed it, RNA. This is the seemingly less famous version of nucleic acids that are really just as important in many ways as DNA, and yet DNA gets all the fame in pop culture. But let's dive right in. And in this video, it's going to be a kind of an intro to RNA, as you can see here. The purpose of it is types that we're going to be talking about that are involved in protein synthesis and the roles of those various types. So to kind of kick things off, what is the purpose of RNA? Why do we need it? Well, nucleic acids, DNA and RNA, work together to store genetic information in DNA and to transmit it, meaning to pass it from parent to offspring, and then once the offspring has it, to be able to express those genes and form proteins and carry out processes and direct development and things like that. So what scientists, biologists have kind of come up with, what they have termed this, a bit of this relationship, one of the most common relationships between DNA and RNA, is known as the central dogma. The central dogma of biology, or more specifically, molecular biology, microbiology. So the central dogma of biology, all right, this is very, very important, okay? Dogma, right? It's a bedrock principle of something, and so central dogma, they made it really, really plain there that it's super important. It's one of the bedrock principles of all of biology, the central dogma of biology, and it goes basically like this. I'm going to very, very much simplify it, that DNA has this relationship with RNA, that the code, the message, the instructions in DNA are transcribed or copied over or put into a version of RNA, which is then used to make proteins. Now, this is not the only function of DNA and RNA per se, but this is one of the main functions of DNA, is that it's going to code for proteins in your body. You have so many different types of proteins in your body that make up everything from hair to nails to skin cells to parts of your bones to your muscles to collagen in your skin. So many proteins. There are enzymes that are proteins. There's just so much. We could not survive and function without proteins. Well, how do they get made? Well, it's this idea here, this central dogma of biology, that DNA is going to be transcribed into RNA, which then will be used to make proteins. And so this video, we're going to talk about this. So let's kind of, that was our quick kind of intro here. We'll dive more into that central dogma in the next video when we actually discuss protein synthesis. But if you want to visualize it in a little bit here, again, we're going to take DNA, the code in it, and transcribe it into RNA, which you'll notice just from the little my simplistic diagrams I've drawn here that it's quite different than DNA, although there are some similarities, and that RNA is going to be used to make a polypeptide. These are amino acids here, right? Each of these is an amino acid. And so I gave them various names and connected them in a random order, so they're not really probably referring to anything real. And a real polypeptide would have many, many, many more amino acids linked in its chain to make the beginnings of a complete protein. But to simplify it down, again, this is the central dogma. All right, the central dogma of biology, that DNA makes RNA, or is used to make RNA, and RNA is used to make polypeptides, proteins. Okay? So let's dive in and kind of look at specifically the RNA now. But before we do that, we have to really start with DNA. And if you recall from our videos in DNA, or anything you know about DNA from previous courses or from watching the Magic School Bus or what have you, then you'll recall that DNA is a double helix, right? We call it a double helix because it's double-stranded. There's two strands, they connect in the middle, and they are twisted. They do rotate in the way they connect because of the molecular structure. So here, if we could kind of take a little section and blow it up, you'll recall that DNA is a long polymer molecule. It's a chain of nucleotides. And so here we have one nucleotide right here. This is one nucleotide. We have a 5-carbon sugar, a phosphate group, and a nitrogenous base, named so because it has a lot of nitrogen. And you'll notice there's 4 different types of those nitrogenous bases represented here. You have adenine, thymine, guanine, and cytosine. Okay, and so there's 4 different letters in this DNA code. And the order in which the letters are placed in these sequences is what will determine the information held within that sequence. Well, how does that actually work? Well, we have to have RNA. RNA is what is going to be used to get this message to the ribosomes where we can make proteins, and without the RNA and without the protein synthesis mechanisms in place, the DNA information wouldn't really be information. It wouldn't be anything. It would just be a string of nucleotides put together with various nitrogenous bases that would code for nothing. So without the mechanisms, without the machinery that's in place in the cell, this would not mean anything. And so RNA is going to be somewhat similar to DNA in that it will also be made of nucleotides. And it will also have a helical structure. But you'll notice that RNA is going to be single-stranded. There's only one side over here. Whereas over here we have a double helix in the DNA, right? So double helix for DNA, single helix for RNA. And you'll also notice, if you begin to study it and look closer, that RNA, well, it doesn't have the exact same nitrogenous bases. Over here in DNA, I have some purple ones, as you can see here, but over here in RNA, there are no purple ones. There are pink nitrogenous bases. So let's look a little closer here. With DNA, here are our nucleotides that are possible. All right, a nucleotide that has guanine, cytosine, adenine, and thymine. Well, in RNA, there are guanine, cytosine, adenine, and this pink one I have here with a U is known as uracil. So RNA already has a major difference there than DNA. It has uracil as a nitrogenous base and no thymine. And this would work similarly when we are making RNA from DNA or when there's interactions between different types of RNA. This uracil will pair up and interact with adenine just like thymine would in DNA. So C and G are going to still hang out together. Okay, they pair up, they match, they have 3 hydrogen bonds that form between them. Adenine and thymine have 2. And you'll also notice the way that I have drawn these, a difference in the 5-carbon sugars. Well, that is because DNA has deoxyribose and RNA has ribose, hence the name ribonucleic acid, RNA. So let's do a little chart here. Let's do a little comparison here with DNA. Again, we said DNA is a double helix. And RNA over here is a single helix. Okay, and then of course we said that the base or the sugars are different here. In DNA, hence the name deoxyribose is the sugar. Over here in RNA, the sugar is ribose. Deoxyribose is called deoxyribose because it is missing an oxygen. Well, really an oxygen and a hydrogen that ribose has. So you have ribose, and then deoxyribose is missing an OH group that ribose has. So those are different, and then we'll find that in DNA you have thymine, and in RNA you have uracil. So if you can remember these three kind of things about them. DNA is a double helix, RNA is a single helix. DNA has deoxyribose, RNA has ribose. DNA has thymine, RNA has uracil. Those are the major differences that we have here that we're going to look at. Now, they do have some things in common. All right, if we look at these three nitrogenous bases, they actually have these in common. Okay, and actually let me write, these are common, as in they share them, they both use them. These are common bases, right? So they both have guanine, they both have cytosine, they both have adenine, but again, that difference is DNA has thymine and RNA has uracil. So if you ever see a sequence and it has T's in it, you automatically know it's DNA. If you ever see a sequence that has U's in it, you automatically know it's RNA. All right, and so those are the big kind of the good intro and comparison of RNA to DNA. Well, let's look though, because this RNA that I have drawn over here on the right, that's not the only type. And there are going to be three, technically more, but three types, major types that are used in protein synthesis. So the first one that I kind of drew, that single helix, that was just a single strand, this is going to be messenger RNA. Whoop, my handwriting needs some work. Messenger RNA, or we call it mRNA. And it's going to do exactly that. It's going to carry the message. So it carries the code from DNA to the ribosome, which I have drawn down here at the bottom, to a ribosome where we will actually make polypeptides. So mRNA is going to carry code from DNA to the ribosome. Okay? Now that brings me to the next type that I want to kind of quickly give you a little information about, and that is ribosomal RNA. Ribosomal RNA. Okay, so with ribosomal RNA, it's exactly what it sounds like. It's RNA that makes up a ribosome. Pardon me. It's RNA that makes up a ribosome. And so what it does, it literally is going to help in the process of protein synthesis. But what it does, it actually makes up part of the ribosome. You'll notice the ribosome here has two subunits, an upper one and a lower one, and these gray sections are some of that ribosomal RNA in there. And ribosomes are also made of proteins. So it's components of ribosomes that play a role in protein synthesis, making those polypeptides. Now I saved the other type for last because it's important to understand the messenger RNA is going to travel to this ribosome carrying the message where it will need to be read and made into a polypeptide. Those instructions carried out, translated, we say. And so one of the ways that occurs, that part of the process, is we use these things called transfer RNA molecules, or tRNA. Now, you'll notice this tRNA molecule is shaped very differently, and this is kind of a common representation of tRNA. That's not really what it looks like entirely, but you'll notice down here these three nitrogenous bases right here, they form what's called an anticodon. And on the messenger RNA, each group of three that it was coded with from DNA is called a codon. The three bases that go together that form part of the code are called a codon. Well, there is a matching complementary, meaning the bases pair up together based on complementary base pairing. There's a matching anticodon on tRNA for a codon on the mRNA. And up here, it is carrying an amino acid that it will use to add to the polypeptide, to make that polypeptide chain. So transfer RNA carries amino acids to the ribosome, and attaches them together based on the instructions that mRNA got from the DNA. And so what we're going to do, I'm going to kind of leave you there. In the next video, we'll go into this process a little more in depth, and we'll come back to this idea of DNA being coded into messenger RNA that's called transcription, and this idea of that messenger RNA going to the ribosomes where it will be used in conjunction with transfer RNA, tRNA, to build a polypeptide. And so, as always, I want to say thank you for watching. I hope that this video helped you understand RNA a little bit better and kind of give you an intro knowledge. I encourage you, like my videos, subscribe, leave me comments, ask for new videos, and as always, thanks for watching.